My name is Aaron Smith and I'm a doctoral student at the Centre for Mathematical Biology at the University of Oxford. In this screencast I'm going to show you that multicellular configurations, known as rosettes, play a crucial role in epithelial cell movements during a process in the mouse embryo called AVE migration. This work has been carried out in collaboration with Dr. Shankar Srinivas and his group in the Department of Physiology, Anatomy and Genetics. The video on screen now demonstrates two simulations, one in which rosettes form on the left and one in which they don't form on the right. Notice the different behaviour between the two panels. We'll come back to this video later in the presentation. The process by which the head end of the body is properly specified and positioned takes place shortly after implantation in both humans and mice. At this stage, the mouse embryo consists of three tissues, shown now in the schematic cross-section on screen. They are the extraembryonic ectoderm in the proximal region, the epiblast in the distal region, and the visceral endoderm, or VE, which is a simple epithelium or monolayer of cells surrounding the other two tissues. The AVE is a special group of VE cells responsible for the correct orientation of the head-to-tail axis. They form at the distal tip of the embryo and show a characteristic migration to the midpoint where the epiblast meets the extraembryonic ectoderm, whilst maintaining contact with the surrounding VE cells and underlying epiblast at all times. Once they reach this midpoint, they start spreading laterally over the epiblast and do not normally migrate onto the extraembryonic ectoderm. A failure of AVE migration leads to abnormal development and embryonic death. AVE migration occurs by intercalation of cells within the epithelial VE. We have observed regional differences in behaviour between VE cells overlying the epiblast and those overlying the extraembryonic ectoderm. While the former region, to which AVE migration is restricted, shows robust cell movement and intercalation, the latter is relatively static and appears non-permissive to migration. Little is known about how cell rearrangement is regulated in epithelial sheets like the VE. In many epithelial tissues, the network of cellular junctions resembles a honeycomb pattern, with three cells meeting at each vertex. Rosettes are a peculiar arrangement whereby 5 or more cells meet at the same vertex. In our recent PLOS biology paper, we show that rosettes also appear in the VE during AVE migration. The video on screen now highlights a rosette forming in the VE. The role of rosettes in the context of AVE migration is unclear. We set out to investigate the significance of rosettes using a combination of experimental intervention and mathematical modelling. We used a vertex model to represent the visceral endoderm of the mouse embryo. In this model, cells are represented as polygons with forces acting on the vertices in the direction shown in the figure. Forces represent cell-cell adhesion, actin-myosin contractility, elasticity, pressure, and protrusion. They depend on cell properties such as those indicated in the diagram. The model was implemented on the surface of an ellipsoid to represent a realistic geometry for the embryo. We simulate rosette formation by allowing pairs of vertices that fall below a certain threshold distance to join together. The number of rosettes in simulations can be manipulated by adjusting this threshold distance. Let's now look again at the video we saw at the beginning of this screencast. In the simulation on the left, rosettes are allowed to form during migration. The AVE migrates as a single, coherent clump of cells. In the simulation on the right, meanwhile, in which rosettes do not occur, the AVE breaks up into distinct clusters, separated by non-AVE cells. Our simulation results thus suggest that whilst rosettes are not critical for migration to occur, they are crucial for migration to occur in the orderly manner observed in wild-type embryos. We were able to validate our computational result by comparing with experiments on mutant embryos in which planar cell polarity signaling is disrupted. In these mutant embryos, fewer rosettes form, and the migration behaviour is indeed similar to that of the simulations without rosettes. Our simulations and experimental results thus show that rosettes are crucial facilitators of ordered AVE migration in the mouse embryo.